Okay, hello class. Today we are going to be picking back up with chapter 9 and the first part of chapter 10. So this lecture that we're going to be starting will serve as both the lecture for this past Friday, which was the 19th of February, and for today, Monday, February 22nd. Chapter 9 is just such a short chapter that I decided to add it into the first part or combine it with the first part of chapter 10. And it's a very, very simple chapter, so we're going to blow right through it. So here we go. We are going to be talking about cell signaling. All right, so cells can signal to each other and they interpret these signals that they receive from other cells and the cell and from themselves, actually. And signals are most often in the form of chemicals. And the same small set of cell signaling mechanisms is going to show up in a diverse range of species and processes. So there are different types of signaling. There's a paracrine signaling, which is a local signaling. That local signaling means that it is a signaling molecule that's going to move through diffusion of the extracellular matrix to nearby cells. So it's signaling other cells that are very, very close in proximity to the cell that's emitting the signal or the signal itself. So endocrine signaling is long distance signaling. And the way I remember this is you have an entire system called the endocrine system, and so you know that it has to cover a very wide range and long distance to travel through the entire endocrine system. So endocrine signaling is long distance signaling. So signal molecules are going to come from distant cells via the circulatory system, and they're going to typically produce a slower response, but have longer lasting effects. So that's a key for endocrine signaling. Now, these are all good definitions to know. You need to know all of these: paracrine, endocrine, autocrine, and direct. So, autocrine signaling is self signaling. The cell actually is producing a signal that comes back on itself and tells it to do something. So, signal molecules can affect the cell that produce them such that the signal and the target cell can become the same or similar to each other. So, say you have an epithelial cell and it signals the epithelial cell that it's directly connected to. Technically, that's considered autocrine signaling, even though that cell is not exactly itself. But we're not going to get that crazy. If I ever ask you, a cell is signaling other cells nearby, always jump to paracrine. That's paracrine, local signaling. If a cell is signaling itself, its direct self, that's autocrine. For our purposes, that's how we're going to define it in a test situation. You're not going to have to be sitting there thinking, well, is the cell really close to it? A same type of cell? I'm not going to try and trick you that way. So direct is just cell to cell signaling. These are the gap junction cell signaling. If it's using it's connected to another cell, and it signals through these gap junctions passing between the cells. That's called direct. Okay, so it's a form of autocrine type signaling, but for our purposes, if one cell is connected to another cell and it sends a signal through a gap junction, direct signaling. Okay, so very, very straightforward. All right, so here it is in an animated version. Autocrine, the cell is going to emit a, a messenger. That messenger comes back around on itself and creates and elicits some response, autocrine signaling. Signaling across those gap junctions is direct signaling. So that this one right here is, they don't spell it out, but this is direct signaling. Paracrine is signaling nearby. They are in close proximity to each other. So that's paracrine signaling. And then endocrine, this is representing using the bloodstream because it's having to go a long distance. So paracrine signaling is a cell signaling that happens over distance through the bloodstream. And another way of saying that is through the circulatory system. Okay. Autocrine signaling example. This is just going to become redundant, but an immune cells signal each other. They will use receptors. to grab the signal. But one immune cell can encounter a pathogen. It's going to produce a signal that converts other immune cells to the appropriate subtype to respond to the pathogen. So that's just an example of autocrine signaling. So direct signaling, this is using those gap junctions, or in the case of plant cells, between the plasmodesmata, which, as we talked about when we discussed cells, plasmodesmata and gap junctions are kind of interchangeable in their purpose. Okay, so paracrine signaling example, the distance between the presynaptic cell and the postsynaptic cell is called the synaptic gap. It's very small and allows for rapid diffusion of the neurotransmitter. And so this is a nearby cell, so paracrine. Enzymes in the synaptic cleft will degrade some types of neurotransmitters, and then that will terminate the signal. So that's paracrine. Here's endocrine signal example. Long distance signaling. Plants and animals use chemicals called hormones, and hormonal signaling in animals is called endocrine signaling. And this is specialized cells releasing hormones that then travel to target cells via the circulatory system or in the bloodstream. The ability of a cell to respond to a signal depends on whether or not it has a receptor that's specific to that signal. So most often target cells are going to need a receptor to be able to grab on to that signal that's coming into it for it to elicit a response. So here are the 3 stages of cell signaling. Reception: the target cell detects a signal molecule that binds to the receptor protein on the cell surface. That's reception. Think of a receiver in football. That football is the signal that's coming in. The receiver has to be able to grab onto it for it to count. Okay, that is reception. The target cell is going to detect the signal by binding that signal to the receptor protein on its cell surface. Transduction is the binding of the signal molecules that alter the receptor and then initiates the signal transduction pathway. Transduction is often going to occur in a series of steps, but this is basically it bringing that signal and eliciting a change in the receptor to continue the signal on down through the membrane and into the cell. That's transduction. And then the response. The response is the transduced signal is then going to trigger a specific response in the target cell. This may need to have gone through a 6, 10, 20 step process of cell signals and then secondary messengers and that kind of thing through the cell, but it will eventually elicit a terminal response of something happening that it was intended to. All right, so this is just an artist rendering of it happening. You got the reception, there's the signal molecule, which we will call the ligand at times, and the ligand will come in and bind to the receptor. Okay, that's reception. And I'll just write that out. That's a ligand, or the signal molecule. We call it a ligand. And then here, the signal molecule elicits a change within the receptor. That receptor change elicits other responses that transduce the signal down. And these are relay molecules, or sometimes called secondary messengers. But that elicits the response to keep going, the signal to keep moving through into the cell. And then after transduction, we have the response. You'll have an activation of some type of cellular response. So reception, this is the ligand we were talking about, is the messenger. The ligand is the messenger, and it will bind to a receptor with very high specificity. And then a shape change or a conformational change occurs within the receptor that elicits an initial transduction of the signal through the membrane. Most signal receptors are going to be plasma membrane proteins of some kind. So external versus internal signal reception. Okay, so sometimes there's not necessarily a receptor. Sometimes the signaling molecule comes through the membrane. So hydrophobic signaling molecules like steroid hormones, like estrogen, testosterone, that kind of thing, they're going to typically diffuse across the plasma membrane, and they're going to interact with intracellular receptors in the cytoplasm. Now, hydrophilic, which is water-loving, or very large molecules can't get through the membrane. They're like neurotransmitters, insulin, and they're going to typically bind to a receptor protein embedded on the outside. So this is an example of a signal molecule here that is hydrophobic because it is able to get through the membrane and bind to an intracellular receptor. So just keep that in mind, that remember we talked about this with membranes. Hydrophobic, all this interior portion of the membrane are these, you got these phospholipid bilayer. And remember that they do not, all this interior middle area does not like water. It cannot be in contact with water. So things that are also hydrophobic, that don't like water, are able to get through. They're like, okay, you're like me, you can come on through, and they'll be able to get through the membrane. So hydrophilic, water loving, has to stay outside in the more aqueous environment, and they'll bind to receptors that are on the cell surface. So you might, if that was a ligand right here, it's going to bind to a cell surface receptor to be able to elicit a response. Okay, so I did a horrible job of drawing, dark ligand, but that is more like what would happen. Very large molecules, hydrophobic molecules, they have to bind to a receptor like that. All right. Transduction, binding of the signal molecule to a receptor, triggers the first step in a chain reaction. The receptor activates another protein, which then activates another and another, and so on until the protein producing the response is finally activated. So at each step, the signal is transduced into a different form, usually a shape change in a protein. Sometimes it can elicit a secondary messenger or molecule to continue down the line. So phosphorylation in transduction is important because this is one way that signals are transduced through phosphorylation. So phosphorylation and dephosphorylation of proteins is a very widespread cellular mechanism for regulating protein activity. So protein kinases, remember we talked about what a kinase is. ACE means enzyme, right? Well, the kine part means it's going to phosphorylate something. So it transfers phosphates from ATP to proteins in a process called phosphorylation. So that's adding a phosphate group to something. So kinases are responsible for adding phosphates. Protein phosphatases remove phosphates. So you need to get this right. Kinases put phosphate groups on, they attach phosphates. Protein phosphatases remove them. So that's very, very key. They rapidly remove the phosphates from a protein. So make sure that you have that straight in your head. And that's dephosphorylation is what that's called. This phosphorylation and dephosphorylation system acts as a molecular switch to turn and activate things either off or on, up or down, whatever is required. And this is just a image. You don't need to know this image here. You don't need to know that. So, but it's just an example of how adding phosphates activates certain proteins. Taking phosphate groups off deactivates proteins, that kind of thing. So, and sometimes it can be in reverse. Taking a phosphate group off can, in some cases, activate things. But most often it's when a phosphate group is added that it's activated. And then the response, ultimately, that signal transduction pathway is going to lead to some type of one or more cellular activity. And the response can occur in the nucleus or the cytoplasm. So it could be something that needed to happen within the nucleus, or it could be something that needed to happen within the cytoplasm. So those signals can create an activation in either area. Many signal pathways are going to regulate the synthesis of enzymes or other proteins, and this is usually done by turning genes on or off in the nucleus. So the final activated molecule in the signaling pathway may function as a transcription factor. And we've talked a lot about the central dogma, which was DNA to RNA to protein. Well, this we found out that the transcription of DNA to RNA and then the translation of RNA to proteins doesn't exactly fully encompass really what's happening to produce the final product of proteins. This transcription factors is really majorly involved, and if you go into further on down into higher levels of biology, into grad school, whatever, you'll learn about this transcription factor processes that in more detail of how genes are actually modified, turned on and off, that kind of thing, for the final result of creating proteins and other necessary uh, molecules. All right, so now let's talk a little bit about apoptosis, because sometimes the cell needs to be told to actually just die. Sometimes it's time for the cells. They've done their job. They've gotten old, and they may be prone to making errors, that kind of thing. So the body has a way of saying, "Hey, we need to signal this cell to die." So cells that are infected, damaged, or at the end of their functional lives are often undergo this programmed cell death, and that's what apoptosis is. It's meant to happen. Programmed cell death is apoptosis. So very, very important. Components of the cell are going to be chopped up and packaged into vesicles that are then digested by different scavenger cells. Um, and apoptosis is going to prevent enzymes from leaking out of a dying cell. You wouldn't want enzymes that break things down to just be free floating in your body, because that would be very hazardous. So they're going to break those down and prevent them from damaging their neighbor cells. Apoptosis also is going to prevent damaged cells from becoming cancer cells, because sometimes cells that are not doing their job correctly could become cancerous, especially if they have something wrong with they're reproducing too rapidly, that kind of thing. So apoptosis is supposed to help prevent that from happening. So, and another example of how programmed cell death is, if you look at the developing fetus, and you look at the hands that are being developed, initially the tissue of the hand is all connected. And then as development continues, the cells between the fingers begin to apoptose. They begin to die off and close off to create individual phalanges of the hand. So that's an example of programmed cell death in apoptosis. So this study guide, very simple. We blew through that. So we're going to jump right into chapter 10 here, which is cell reproduction. Now, cell reproduction is going to get a little bit more complicated, but we're going to do the first 10 slides or so. Now, all cells divide. Okay, so this is how cells reproduce. Multicellular organisms are going to use this cell division for growth, maintenance, and even the repair of cells and tissues. So single-celled organisms use cell division to actually reproduce. Okay, so they're reproducing completely themselves, and this is the happening through both asexual reproduction in cells and then in just growth development. This is just an example in some microscopic imaging of cells going through an asexual reproduction and growth development, which could be an example of like tissue renewal. So like your epithelial cells that are constantly shedding off and that kind of thing on your skin. And so they're constantly going through this growth and development stage where they're renewing by doing the cellular division. So sorry for my slides again. I don't know why it keeps doing this, but your slides shouldn't be overlaid like this with the words over the titles if you download them directly into PowerPoint. Okay, so genome, it's a cell's complete genetic information. So that's a good definition right there. What's a genome? A cell's complete genetic information, or an organism's complete genetic information. In prokaryotes, the genome consists of one double-stranded circular DNA molecule. So prokaryotes, one double-stranded circular DNA molecule, and it resides within the nucleoid. Very important. All of this is very important. That looks like a good test question. In eukaryotes, the genome consists of several double-stranded DNA molecules in the form of chromosomes, okay? And those are going to be found within the nucleus. So eukaryotic chromosomes, the number of DNA molecules or chromosomes in the cell number varies among species. Within a species, the chromosome number is consistent but it may vary with developmental stages or specific cell type. So this is an example of a chromosome. It's made up of two sister chromatids. So you have one chromatid here and then one chromatid here. And so these are sister homologous chromatids, and they connect right here through the middle at the what we call the centromere, and there's a centromere on each sister chromatid. So I kind of did that a little. There's one centromere on each sister chromatid, and that we call that the centrosome where they connect. So eukaryotic cell types. Okay, so there's two types of eukaryotic cells. There's somatic, which are cells that typically have two matched set of chromosomes, and which makes them diploid. So that would be diploid is just 2 to the n. And then some somatic cells divide, most somatic cells actually divide as an organism grows or when damaged tissue needs to be replaced. Okay, so that's like your epithelial cells and different mucus cells and that are in your that are lining your internal organs and that kind of thing. And then you also have what are called gametes, and these are the sex cells like eggs and sperm, and they're going to have half the number of chromosomes, so they're considered haploid. And this is important for reproduction because gametes fuse during fertilization to form diploid zygotes that then develop into organisms that are made of somatic cells. So that's why they only have one, so that they fuse together to form a diploid zygote. So let's talk about some eukaryotic cell types. So eukaryotic DNA must be condensed into compact chromosomes to be able to fit in the nucleus because it's so immense. Like there's just so much information. So they get condensed down and compacted into these chromosomes. So when you look at this chromosome, you can see it's very, very packed in and condensed. Like these are all DNA strands and molecules within this chromosome. So all this is just super tightly packed DNA. All right, that's what a chromosome is. And then so you can see that this is what a DNA strand looks like here. And the DNA is wrapped around histones. Okay, and histones are what help keep it really packed. They're these little orange things, if you will. I don't exactly know how to describe that color, orangish yellow, whatever. And then you can see the DNA gets wrapped around them, and that's what helps compact it down. And then you have this further condensation of the chromatin all around within the chromosome. And then so if you look, this tightly packed thing is just in one area of the chromosome, and it's like that all the way through. So that's how packed chromosomes get. And to be able to reproduce, the DNA has to become uncompressed so that it can be read and then transcribed into RNA, and then that RNA is then translated into proteins, and then those proteins become cells, and then the cell cycle is going to actually look at this process of actually creating additional daughter cells. So we're going to talk a little bit about that. So what's happened is most cells, as you look at a cell, it's going to be most often found in either this interphase portion here or the interphase portion here. So you have G1 and S phase. Those are the biggest portions of a cell's life. It's either going to be growing itself or it'll become in the part where it starts to synthesize the daughter cells. So you have G1, the S phase, then you have G2, which is an additional cell growth stage, and then you come into the reproduction stage, this little sliver. It happens very quickly because you can see it's only that much of the cell's life. And it is broken down into this mitotic phase of mitosis and then cytokinesis here, and that ends up forming two daughter cells. All right, so two major phases here. The interphase, which is in blue, is the time for normal growth and preparation of cell division. And then the mitotic phase in red is where the replication of the DNA and cytoplasm is happening, and then it's split, and then the cell divides, and it becomes two daughter cells. Basically, you start out with one cell here. It goes around, and once it hits this, it starts to elongate, it fissures, and before it jumps out, it becomes two. So you started with one, you ended with two. All right, so that's what it's saying is happening here. So, all right, so we are going to stop right here before we jump a little more into a deep dive into cellular replication, and we'll start back up on Wednesday of this week. So, be doing chapters 9 and 10 homework quizzes today, if possible. You want to really jump on that. Since we didn't have a lecture this past Friday, I kind of gave y'all the day off. Want to go ahead and get back into it now and attempt 9 and 10 today. All right. Have a great week.